focused on the bacterial genetic mechanism of transformation, a form of horizontal gene transfer, particularly relevant in the context of antibiotic resistance. Each includes 5 answer choices and detailed rationale. Question 1. Mechanism of antibiotic resistance acquisition. A researcher is studying a strain of Streptococcus pneumoniae that was initially sensitive to penicillin. After incubating the bacteria with lysed bacterial fragments from a penicillin-resistant strain, the S. pneumonia becomes resistant. Which of the following best explains the mechanism by which resistance was acquired? A. Transduction by bacteriophage. B. Conjugation via F+ plasmid. C. Transformation with naked DNA. D. Transposition of resistance genes. E. Spontaneous mutation. Correct answer, C. Transformation with naked DNA. Explanation? Transformation is the uptake of naked DNA fragments from lysed bacteria into a competent recipient cell. S. pneumoniae is naturally competent and can incorporate exogenous resistance genes through this method. A. Transduction involves phage-mediated gene transfer. B. Conjugation requires direct cell-to-cell contact in plasmids. D. Transposition is movement of DNA within a genome, not between bacteria. E. Mutation is not responsible here. Resistance came from another organism. Question 2. Experimental inhibition of transformation. In a lab experiment, a scientist incubates Neisseria gonorrhoeae with DNA from a rifampin-resistant strain of Neisseria meningitis. The gonorrhoeae strain becomes rifampin-resistant. In a repeat experiment, the scientist adds deoxyribonuclease DNA to the mixture and finds no resistant colonies. What conclusion can be drawn? A. Resistance was acquired by transduction. Conjugation occurred between the strains. C. Transformation was prevented by DNase. D. Transduction was enhanced by the enzyme. Resistance was caused by point mutation. Correct answer. Transformation was prevented by DNase. Explanation. DNase degrades free, naked DNA, thereby blocking transformation. Since no resistance occurred after DNase addition, the original transfer must have happened via transformation. A and D, transduction is not affected by DNase since phage DNA is protected inside viral particles. B, conjugation is plasmid-mediated and does not involve naked DNA. E, mutation would not be prevented by DNase. Would you like